Welcome everybody. Thank you, Bert, for the kind invitation to give this seminar. It was planned differently. It was planned in June this year when I was attending or planned to attend the Gordon Research Conference on forensic DNA analysis nearby Boston. And I thought I can combine also visiting Henning Thiemeyer, which is a long friend, saying hello to Bert. But that all was canceled because of Corona. And then later on, I was asked to perhaps still give the seminar, but in this way, which I'm happy to do. So the idea was to tell you a little bit about the genetics that make us looking as we are, and then focus not so much about how to find the genes, what the genes do, but how one can use the DNA information in those genes to predict how we look. And there are certain applications. I will not go really much into those. One is in forensic, one is in anthropology. I give one example each at the end. So the idea really is, because you folks are epidemiologists, public health people, how you can use DNA to predict how we look. So as you all know, and I would normally say now, if you look around in the seminar room, which I can't, so you probably look, you're sitting all at home and can't look at anybody. But there's, of course, enormous variation in human appearance. And this is partly substructured for different continents, but of course, within continents, there's also enormous variation. So basically, everybody looks differently. And of course, we know that there are some exceptions. There are unrelated people, so-called look-alike, that look the same. And if we would understand all the genetics, we would also know why, but we don't. But besides those, which are really rare, it basically is these type of people here, namely monozygotic twins that look identical. And because they look identical and they almost share all of their DNA, as we know that genetics plays a role in human appearance. And you can quantify this by calculating heritability, and you will see that all the appearance traits have very high heritability. They are not all completely genetics. Depending on the trait, you have environmental factors, but the genetic component, the heritability for all of these traits is very high and is much higher than the environmental component. And that's also the reason why monozygotic twins look the same, at least for the younger period of age. And then when they become older, and one is an office worker and never gets the sun in his face, and the other is a street worker and gets lots of sun in his face, then the one will show more wrinkles with advanced age. That's the environment, but they will still look very much the same because it's genetics. So I show you examples of appearance traits where we understand more of the genetics and therefore can also utilize the DNA information to predict the trait. And that's I will start with. And then I come to traits where we don't yet know so much of the genetics, and therefore, as you may imagine, can predict those traits much less good. And at the end, I show you some examples. So eye color is the trait where we probably understand most. And this is not because we know all the genes, by far not. You will see that in a minute. But there are some of these many genes that have a larger effect than others. And the idea has existed in the past that there's only one or two eye color genes. And as you may know, eye color as a phenotype even was used in paternity testing, with the idea that as long as there's one brown eye color parent, the child has to be brown. And if not, then there was the paternity questioned. So this idea is long gone. We now know that there are many eye color genes. And we also see in those genome-wide association studies that some of those genes have a much higher association value than others. And also the beta value, so if you quantify the effects, for some of these genes, these effects are much higher than for others. And that's the reason why we can actually predict eye color just from a few genes, as you see here. So here you see accumulative AUC values, right? Area under the curve, which is calculated from sensitivity and specificity, as you all know better than I do. And this is done in a way from the right to the left by increasing the number of DNA markers, single nucleotide polymorphisms. And already with the first one, if you know what the first one is, as you see here, you get an AUC that's close to 0.9, where you all know that an AUC of 1 means every time you predict in these 6,000 Dutch Europeans, you would be right. So with one SNP, you're already at 0.9, because this SNP has a quite large effect. And we now know from our functional studies that this basically is an enhancer that works like a switch between pigmentation and non-pigmentation. But you also see that this doesn't really work so good for the non-blue, non-brown eye colors, because there the AUC is much lower. And you then add SNPs from different genes, you see that the AUC increases, but you also see that it doesn't increase very much. So many years ago, we wanted to have a very simple test for eye color, and we took 6 SNPs from 6 genes, and you may bet that these were the first here in this chart. And you can see on the left-hand side that even if you increase the number of individuals in your prediction models to more than 9,000, you basically remain with about the same value. So 0.945 AUC for blue and brown, and about 0.74 for intermediate. We do not yet completely understand why that is. It could simply be that intermediate has much more different phenotypes than blue and brown, because you basically are in the middle of the spectrum. And not that there are specific genes that make the eye color non-blue and non-brown, namely intermediate. But this is not yet worked out completely. So then, because we wanted a tool that we can apply, for instance, in forensics, we develop a test. So that's a lab test, which may not be so interesting here. The good part here, it's quite sensitive. So you only need 30 or so picograms, which is not much, to get the test working. And that's important in applications like forensics, where you don't have much DNA. And then, of course, you don't only need a DNA test, you also need a statistical model, because you have to put those genotypes into the model, and that's a web interface we developed. And then you press a button, and what you get is probabilities for blue, brown, intermediate. And that's then, of course, by using the model's data, thousands of individuals with genotypes and phenotypes, but then for your sample for which you put in the genotypes, you're getting individual probabilities. And those, of course, you can use in all types of application when you ask, what is the eye color of that person, if you only have a DNA sample or you have a bone where you don't see the eye color, but you get DNA, and then you can predict it. So this is how the test works. Those eyes are ordered as the test outcome orders them, with the highest probability for blue on the upper left corner, and the highest probability for brown in the lower right corner. And then you have decreasing blue eye probabilities going down from the upper left, and you have decreasing brown eye probabilities going up from the lower right. And as you see here, there's one eye circled in red, and that's the one from these 40 from the department and across the floor, where you have a true error, because you have a 96% probability for brown eye. As you can see, the eye is not brown. We know these people exist, and we had one in the department, so therefore we knew for sure. But we also know from these very high AUC values for blue and brown that these are very rare. Obviously, you have varying probabilities for blue eye colors that look the same. In this case, all the 2 left columns, it varies from 78 to from 87 to 97, but it's all very high blue eye probabilities. So here's an example when you do eye color prediction around the world and DNA sample of 1,000 people from 50 populations, and you see the predicted eye colors, and you also see that in those areas where you only expect brown eye people, Asia, Africa, Native America, Oceania, you only see black brown eye predicted people. And in those areas where you expect eye color variation, you see predicted eye color variation. We don't have the eye colors of the people, but of course we know how eye color is distributed in a worldwide way, and that gives us an idea how accurate this can be. Normally, of course, you do those tests by having individuals for which you know the eye color. So is that it? Can you find more genes? Yes, you can. And those of you that know about genetic epidemiology, there are typically 2 ways how you can improve your genome-wide association testing. One is you use more detailed phenotypes. And that's what you see here, because that's a GWAS on continuous eye color in 9,000 or so individuals. And if you would do a GWAS on categorical eye color in these individuals, you would see these 5 genes here in blue. And then when you do continuous eye color, because you have more phenotype information, actually in this study we showed that you can find additional genes, novel genes, just because you have more phenotype power. And then the other way, which is the typical way, as you may know, how to improve your GWAS results is you brute force increase the number of individuals, the sample size in your GWAS. And we also did that for eye color, and that's a paper that's soon coming out in Science Advances, where we gathered data from all types of sources, including 23andMe on your side of the world, and doing GWAS in almost 200,000 people and found 50 genes. So this is a trait where people thought in the past has only 1 or 2 genes. Yeah? So it's highly complex genetics with lots of genes that have small effects, but there are few genes with larger effects. And if you use those few genes with larger effects, you can predict at least blue and brown eye color, but not the non-blue brown. So what about hair color? Obviously, we know there must be a link because the phenotypes are linked, right? We all talk about Europeans when we talk about eye color variation, hair color variation. There's only one exception in the world where there is hair color variation without European admixture. I'm not telling that now, so you may think about it, and perhaps you can ask me later on. But otherwise, eye and hair color variation, in contrast to skin color variation, really is a European phenomenon, right? And as you know, there is a relationship. Many blondes are blue-eyed, right? So the more north you go, the more blonde, the more blue eyes. The more south in Europe you go, the more brown eyes, black hair. And therefore, as you would expect, the genes also overlap, right? They're not overlapping completely. So there's one important exception, which is here. I don't know if you see my cursor. So it's the important MC1R gene that is so far the major, if not the only gene that makes hair red. Yeah, so red hair is not a polygenic trait in contrast to all other hair colors. And that gene, MC1R, seems to be not involved at all in eye color. We don't know why, but it is involved in skin color, as you may know, because redhead individuals typically have very light skin. So it makes light skin and red hair. But otherwise, it's all the same genes. And the reason why we partly use different variants in these genes has to do with association effects that we partly don't understand because we don't have the functional ones. So here is the first study we did to predict hair color. At that time, we only had 400 Polish Europeans that even had to be enriched for red hairs, because normally in 400, if you have normal frequencies, you hardly have any red hairs because red hair is very rare. Even in Ireland, where it's more frequent, but outside of Ireland, even more rare. And as you can see, we can predict red hair very accurately. Why? Because many of these 22 SNPs here are from that MC1R gene, and they are from that gene by purpose, because we know they allow to predict red hair color, because there's no other gene that is involved in red hair color. Whereas about half of the other SNPs then, which were picked from hair color association testing, are associated with other hair color, and therefore they allow predicting other hair color, although not as good as you can see here, right? So AUC 85 or 0.85 for black hair, and least accurate for blonde and brown. And you may think for a minute before I tell you why DNA-based prediction of hair color would be least accurate for blonde and brown compared to black and red. So I tell you in 5 seconds. So we made a test, which we called HiRisplex, which can do eye and hair color together because the SNPs partly overlap. We made a lab test because we want to use it, highly sensitive, not as sensitive, because it has more of these SNPs, more of these DNA variants, and the more of these variants are on the test, typically the less sensitive it is. And there's a website where you plug in the genotypes of these 24 SNPs and you're getting eye and hair color probabilities for your particular application. And here are the examples, and then I tell you in a minute why it's least accurate for blonde and brown. Individuals with very high probability for black on the left side, then for brown, and then for red. And you see that all the colors are red. So these images were not selected based on the phenotypes. They were selected based on the test outcomes. And then you see these individuals here, which have a fairly high probability for blonde and are brown and are blonde. And you see next to them individuals with brown hair color, and they still predict highly for blonde. Why? Because these people were blonde as children. as I was, and I predict brown, even though I was blonde as a child. But some people that become brown in adulthood predict blonde as they have been as a child. And we don't know why that is. And we also don't know why some people don't change from blonde to brown and stay blonde and predict blonde. So there's lots of things that has most likely to do with gene regulation and nothing really with coding SNPs, but rather with regulating our genes that makes this phenomenon of age-dependent hair color darkening. And of course, we have another phenomenon Bert can tell us more about, and that's basically age-dependent loss of hair color, which then kicks in typically at a later age. And we also don't know much about that. I didn't include it here, but we only know one gene, which actually is a hair color gene that pops up in a GWAS on gray hair. There must be many more genes which we don't know, but I think the complication here again is it's a gene regulation, right? So the genes for pigmentation are shut off with lifetime, and that perhaps is more difficult to find in a simple GWAS approach. Okay, how you find more genes? Well, brute force enlargement of GWAS, as we did here in the UK Biobank, 23andMe, the Visagen Consortium, which I'm co-sharing together with Tim Spector. 300,000 people gives 124 genes for hair color. Again, this is a trait where people thought in the past, oh, this can't be have many genes, right? It's hyper multiple genomics thing, right? So people have not expected that. Of course, many of these genes have small effects. Many of these genes actually were novel at that time and still are, right? So people had no idea that they would be involved in pigmentation. And if you actually use them for prediction, which we did in this study, you can see in red here with the new model that indeed they increase prediction, which you expect. But as you see on top here, this is using 260 SNPs, whereas in our simple test, we just used 22 SNPs, right? And with that ratio of using 10 times more SNPs, these increases in prediction accuracy are not so impressive, right? Obviously, if you want to predict and you want to predict as accurate as you want any increase that you can, but there's a problem if you do DNA testing, for instance, forensics, to type 260 SNPs, because from a few cells, this is not really trivial. You know? So there's an issue here to develop DNA technology that can utilize hundreds of SNPs in a simple and sensitive DNA test. Okay, and then skin color, you may wait for that, which of course is the pigmentation trait that has a global distribution, yeah, and that causes a big trouble for GWAS, as you all know, right? Because if you would maximize in your GWAS for phenotype, you would have to pool Asians, Europeans, and Africans, because that's the maximized phenotype variation of skin color. But of course, you can't do this because you're getting lots of false positive, namely lots of other genes that are different between the continental groups that have nothing to do with skin color, right? So what people do, therefore, is doing skin color GWAS in Europeans. But there, of course, you have only a fraction of the phenotype variation, and therefore you will never see all the genes, right? So therefore, the gene list we have exists, but is by far less complete than for eye and hair color, because eye and hair color are European traits. So can you then predict? Well, we're trying to do that. It works. Surprisingly for us or not, if you think what pigmentation variation is and where our origin is, namely in Africa. So we all were dark skin in the past. So therefore, the dark alleles are ancestral and the light alleles are derived, because humans left Africa, they became lighter, and that's genetically controlled, right? So therefore, with SNPs that come from European GWAS, we can predict dark skin color much better than light skin color, because it's the ancestral allele, right? And we haven't yet found enough of these light-derived alleles to predict the light colors. At least if we treat the light colors in 3 categories as it's done here, because that would quantify, that would allow getting an idea about European skin color, which comes at least in 3 different color categories, where if you pool them, your prediction accuracy would be much higher. But then you would say all Europeans have the same skin color, which certainly we know is not true. Yeah? So therefore, these accuracies here for the 3 light colors are lower than for the 2 dark colors. And here you see the first attempt to a test. So these skin images, they are ordered in the left upper for the highest probability for the very pale category. And then if you go all through the lines to the right lower corner, you end up with a skin sample with the highest probability for the dark to black category. So what you should see is going from very light to dark through the lines, and it's getting darker if you go from upper left to lower right, which I think you see, but it's not as accurate yet, especially for the 3 light colors as you would like to have it. And that has to do with our not yet so good understanding how skin color genetics works. Same business, test, website, distribution. Here's an example. You may guess who that is. A person with 55% brown hair color, but 40% blonde hair color. So you see there's something going on there. 97% blue eye color, 66% pale, and 24% intermediate skin color. And that's me. Right, and again, I was white blonde as a kid. Yeah. So what do you do to help this? Well, obviously you enlarge your GWAS, which we did a couple of years ago with the UK Biobank and some other samples. It wasn't so successful as our large GWASs for eye and hair color, where again, one reason is that the phenotype variation in Europeans, and this is basically all Europeans, isn't that large. And also that the quantification of skin color, especially in the UK Biobank, is not so sophisticated. It's basically questionnaires on skin sensitivity to sun. Yeah, so it's tanning ability, which of course is skin color related, but is not necessarily only skin color. So you would need the complete Fitzpatrick scale, for instance, to derive skin color, which I don't think has been done there. At least it's a GWAS on tanning ability. At least it gave new loci, also for pigmentation, because of the enlarged sample size. Okay, so that's the bit on those things we understand most, not that we necessarily know all the genes, but because there are some genes with larger effect. And that phenomenon, as far as we know, does not seem to exist for any other human appearance traits, which are also highly multigenic, but they do not seem to have these large effect genes. And of course, that makes it problematic for prediction, because that means you basically have to find all these small effect genes before you can predict anything, because you don't have any large effect genes where you only have to find 1 or 6. And that's a dilemma, at least when it comes to the non-pigmentation traits. You see here one, and there's another, actually I forgot as I now see, which are still pigmentation traits, which are emerging. One is eyebrow color, which is a pigmentation trait. That's the color of your eyebrows. And the other pigmentation trait, which I forgot I see, is freckles, yeah, which is a pigmentation trait, which is largely linked with red hair and light skin. But there actually is not MC1R, it's also other genes kicking in, where there is a prediction model now for freckles. But that, again, takes advantage of the larger effect of size phenomenon of pigmentation. So here's eyebrow color. Nobody did an eyebrow color GWAS before. We had the data and did it, right? But also because we had an interest in enlarging the number of traits you can predict for certain applications. And also there we know that there's a large correlation with the phenotypes, right? So many dark hair, dark eye people have dark eyebrows, and a lot of blonde hair, blue eye people have blonde eyebrows, but there are quite a number of blonde hair, blue eye people with dark eyebrows. I was one of them already when I was a child and had blonde hair. I had darker eyebrows, yeah? And anecdotal knowledge, and I don't have the numbers, would say, well, perhaps there you could have seen that you will be the one changing into brown because you never had white eyebrows. Yeah? So that's the working hypothesis. Perhaps someone at some time has the data to look this up. Yeah? But therefore, there is a correlation again, as between hair and eye, but it's not a complete correlation, right? And therefore, we found pigmentation genes, but at least we found also one gene that appears to be a novel gene here, at least based on that GWAS threshold, which hasn't been popping up before in eye, hair, and skin color GWASs. So how well does it predict? Well, as you see here with 25 SNPs, you're getting AUC values now I can't see this because I have, here, I can move that bar. 0.62, 0.67, 0.7, yeah, so not as high as we want them, As a reminder, as you all probably know, 0.5 AUC means random prediction, right? It's like tossing a coin. So also it would be using no DNA information. So basically we're not even yet in the middle between random prediction and completely accurate prediction with this. So now we leave the pigmentation area, yeah, and now it becomes complicated because apparently we deal with multigenic traits with small effects for basically all the genes people see. One of those is head hair structure. There is still a little bit of a difference between effect size, yeah, and that's why you see larger GWAS still popping up with more genes. But the magnitude of effect size difference is much less than what we know from pigmentation. So here we did a GWAS on a large sample size. There was a GWAS before on Europeans that fought one gene, TCHH. This then with more individuals finds 12 loci. At that time, people found 3 others, so there were 8 novel. So there are a number of genes for hair structure. Well, this is actually right hair morphology. So if the hair is straight or curvy or wavy or frizzy, or for frizzy hair, typically is only in Africans and New Guineans and Australians. It does not exist in Europeans. So there it's curly. And apparently there are a number of genes that cause that phenotype. Can you predict it? Well, not yet that good, as you see here, right? So with 30 SNPs in this model, 2,500 people, AUC of not even 0.7. On the other hand, if you look down, you see the non-Europeans here, there the AUC is almost 0.8. You know, so there's a huge difference. Why is that? Because there's one gene actually that makes straight hair in Asians, has quite a large effect, and that variant does not exist at all in Europeans. You know, so you have straight hair in Europeans and you have straight hair in Asians, but the straight hair in these 2 continental groups seem to have at least a partial, and for that gene, a quite large, different genetic makeup. You know, so that's an example where the same phenotype, what you think is the same phenotype, namely straight hair, may not necessarily be caused by the same genes. And we also have the other example, because there also is a gene that makes European hair straight, and that gene doesn't exist with that DNA variant. Well, the gene does, but not that DNA variant in Asians with straight hair. You know, so there's a difference here for straight hair. And because that Asian gene effect is higher, we see a better prediction if we have that SNP in the model for non-Europeans, particularly Asians. There's lots of Asians in this non-European data set than what we see in Europeans. So hair loss, of course, is a famous one, right? So people, I just gave a lecture yesterday and then I got the question, there is anecdotal knowledge saying that one should not look at his father when wanting to find out if one gets bald with age, but at the father of your mother. So I was actually surprised that that student had anecdotal knowledge. And if that is anecdotal knowledge, then it is really genetically verified, because the gene, and again, there are lots of genes involved in hair loss, with the largest of all these small effects is indeed on the X chromosome, which of course the males get from their mother. And therefore, if they look at their father, they don't look at the X chromosome, right? But of course, if they look at their mother's father, they're looking at their genetic predisposition on the X chromosome that makes them being bald, right? So in that respect, this anecdotal knowledge of that student was indeed true, but it was also not true, because actually there are probably dozens, if not hundreds of genes that make men bald. And this was a time when we took the available knowledge from the GWASs and asked, so can we predict? Well, that was 2015, 5 years ago. Now the knowledge is very different, because large-scale GWASs were done on male pattern baldness. So at that time, again, the AUC was just not even in the middle between random and completely accurate, with 11 or 14 SNPs. And at that time, we couldn't really enrich the number of SNPs because there were not more genes. And of course, if you then take too many SNPs from the same genetic loci, you can add noise, and then your prediction can go down. So this has changed. This is one of those more recent GWASs in the UK Biobank data on male pattern baldness. There was another one then not on a population based, but on a case control based, involving lots of these early onset baldness men. And that also is an interesting question, right? So is that the same phenotype? Is that the same genetics if you get bald with 20 or with 60, right? And there's lots of overlap between the outcomes from these different GWASs, population-based and case control. So it really seems that they are the major genes are involved in both. And the other question then is, why do some people get bald with 20 and others at 60? So again, that may be a gene regulation effect, which is more difficult to find out. Yeah, so here in 40,000 UK Biobank, they found 250 genes, right? So enormous number of genes for baldness. 200 were novel, some were actually overlapping with the large GWAS in parallel, and now there are 2 or 3 or 4 even on male pattern baldness. And they actually used this also for prediction by splitting the data, so doing a little bit of data cosmetics here, but of course, not really, because the DNA markers they use for prediction, they basically pick from the same data set, which is not really accurate. So these AUC values, as you see below here, they appear very high, right? So AUC of 0.78 between severe hair loss and no hair loss. But unfortunately, these people, they used a trick because they left out individuals that were not severe and not no hair loss in this analysis, right? Because they did an AUC not for all per category, but only comparing these 2 categories. And then, of course, when you leave out data from your prediction, you're not doing something right, because in a real population prediction, you have those other individuals with the slight and moderate hair loss also there, right, which you don't have in your prediction model. So therefore, it may easily be that these values are overestimated. But again, as you see here, you need hundreds of SNPs for this. Why? Because the effect size is so small. So body height, speaking about small effect size, you probably know the story from the GIANT consortium, right? Before 2010, there were a couple of papers with a couple of thousand individual GWASs finding 1 or 2 or 10 genes. So then they're all united in the GIANT consortium. This was their first body height GWAS. And actually, because of those studies, body height is the appearance trait where we know most about the genetics. Not that we understand most, but we know most. Why? Because people can do GWAS in 180,000 or 200,000 or 300,000 people. And they could do this already years ago because people united in this GIANT consortium. So 180,000 people gives you 180 genes, 180 genome-wide significantly associated loci, right? And how much do they explain of the body height variation? 10%. How much is heritable of body height? 80%, right? So you do a GWAS in 180,000 people and you miss 70% of what should be there. Well, this tells you how frustrating GWAS on multigenic traits with small effect size is, right? Obviously, you can ask, well, let's do a shortcut. Let's try to predict normal height, but extremely tall people. And if you work in the Netherlands, you can easily do this because there are lots of extremely tall people. So this is what we did at that time, predicting tall people. And as you see, if you do this, you actually get an AUC that is in the middle between random and completely accurate. Yeah? But of course, if you would do normal height prediction, you would never be able to do this with only 180 or so SNPs. So then GIANT continued, as you probably know, right? 250,000 people, paper 2014, almost 700 significant, independently significant genes or SNPs. What do they explain? 16% of the height variance. Right? How does that translate to tall statue prediction? Well, it's getting there. It's 0.8 for extremely tall people, but you need 700 SNPs, right? And that's a problem for certain applications, as in forensics, where you can't really type 700 SNPs because your nice microarrays, which you're using in these studies, they will not work on a crime scene DNA. And they will also not work on a DNA you get from a bone sample because you want to know how tall the person was. Well, for body height, you don't because you can measure the skeleton, but for eye and hair color, it's different. So finally, what about the face? And you are operating in a country where there is a company that makes business with police to predict facial variation from DNA. We can discuss that later, but I hope from the few slides I put in here, you will guess that this can't be true. And this company doesn't publish anything and doesn't tell anybody how they do this and what they do and how they validated what they do, but they simply say, we can. So can they really? Well, so there are a few GWAS studies on facial shape. We have done 2. Here you see the latest in 10,000 Europeans. You may ask, why not more? Well, it's not so easy to do a GWAS on facial shape, because first you have to phenotype your face, right? Which we do in digital images. We develop software that can characterize these landmarks. You can also do it non-landmark based. So this is not so easy than a simple questionnaire for eye color, right? So lots of work. If you do this, indeed, you find genes. You don't find many genes, but you also don't use many individuals, right? 10,000 people is not 100,000. At least 24 genome-wide significant loci, 17 of them are novel. Different parts of the face, as you see here with these lines. So these colored lines, they correspond to the colored lines and the genes in the Manhattan plot, right? So there's different genes for different facial distances. In this study, we had 13 landmarks, because we used different studies that had done different phenotyping. So we only had 13 landmarks overlapping, and from those 13 landmarks, you can calculate 78 distances, and then we did the GWAS on those distances. Yeah. How much do they explain? Well, this is the frustrating part, and in a way, that's completely expected for a complex trait as the human face, because these SNPs, they don't explain much variation of the face, right? So per SNP, and these are those from the genome-wide significant loci, they explain less than 1%. And if you take all of them together, you do regression and you pick 31 SNPs from those 24 loci, they together explain not even 5%, right? And if you have something where genetics explains 5%, I guess you will agree that's very hard to predict a face from that information. Of course, what this company likely does, it doesn't do genetic prediction, it doesn't look for the genes, it simply does genomic prediction in whatever machine learning model, but that is even more critical. And if you want to discuss that, we can do that later. Okay, so now I show you these 2 examples, and if you want, we can also discuss more. So this is one example for DNA prediction application anthropology or human history. You may have heard that story. 2014, King Richard III was digged up underneath a parking lot in Leicester City. So this was the last king of England who was killed in battlefield. Yeah? So if you know Shakespeare, then you know King Richard III. He lived in the 15th century. He was killed by someone who wanted the crown, and then the crown changed from this family to another family, right? So he was found. His feet were not, because his feet were missing, because there was a building from the Victorian times. And if these builders would have moved their building 2 meters to the right, King Richard III would have been gone, right? So it was such a luck that he was buried, of course, in a former church, which doesn't exist anymore. And there was a big project to find out where the church was, where the grave in the church was. And then they saw, wow, this is a parking lot. How lucky are we? And they dig trenches, and in the first trench, they found a skeleton with scoliosis, and they said, wow, this can't be him. But of course, he was described by Shakespeare as a hunchback, right? So then they did DNA testing, and they identified maternal relatives living nowadays, being related on the maternal line with King Richard III, who lived in the 15th century. Imagine that this exists in the records, and one of them knew it and the other didn't. And one of them matched the complete mitochondrial genome, which you may know is maternally inherited. So all the more than 16,000 base pairs in mtDNA matched between someone living today and this king who lived in the 15th century. And the other person they digged up in the archive had a 1 base pair difference from these more than 16,000 mitochondrial DNA base pairs, right? So they identified him by DNA as well. And we were involved in eye and hair color prediction. So here you see the values. The markers were done from the bone in a very different way, because it's ancient DNA, right? It doesn't really preserve as nice as a crime scene blood stain we are typically working with. These are individuals we know that have very similar high probabilities for blue and brown. And as you may expect already, some are blonde and some are not blonde, but have been blonde as child, right? And they still have a high probability for blonde. So what you would conclude then, even you would, I guess, is that King Richard III most likely had blue eyes with a 96% probability, very likely, and blonde, or if he was like me, but not because I don't have such a high probability for blonde, who changed in adulthood from blonde to brown, he may have been brown as adult, right? Because typically the king is then not a child. In this case, he wasn't. And then you may say, well, why don't you look at paintings, right? He was a king. There must be paintings. Indeed, there are paintings. And those paintings show him with all types of eye colors and all types of hair colors. Why? Because all those paintings, even for a king, were done post-mortem. And then it was the artist's memory. And this one here was done closest after his death, and indeed shows him with light eyes and light brown hair. So probably he was one of those that darkened with age, and therefore he has such a high probability for blonde. Right? Whereas if you look at later paintings, you see him with black eyes, you see him with black hair, you see all types of things. And the other one is this here, and I can't really show you examples, but as you may imagine, those things are quite interesting for forensics. Why? Because DNA identification only works if you know already the person you want to identify, because you need to have the DNA profile, because you do a comparative analysis. DNA from crime scene compared to DNA of a known person. If there is a match, you have identified the person. If you do not have the DNA profile of the person you want to identify, that's it. It goes to the drawer, and the case is a cold case. So imagine if you can describe how a person looks like, and this is what we can do at this moment, but in the near future, very likely, as you saw in the second part of the lecture, we can do more. We can predict eye, hair, and skin color. We can also predict geographic origin. We can even predict age with an error from DNA. Some companies say they can predict the face. I don't believe this. So obviously, if you can do this, and you have such a case, a cold case, no DNA match to anybody you have in your forensic DNA database, you can imagine that this could be useful information for the police to find the person. So this is unlikely to lead to a single individual, but it's likely to lead to a group of individuals that match your predicted appearance, age, and ancestry information. And then your DNA profiling can be done on a restricted group. So that's at least the idea. That's practiced, for instance, in the Netherlands. They have all the legal requirements for this. They do another trick to combine this. If there's questions, we can discuss this. And they are solving cold cases, actually, by using this information as investigative tool, as the police uses all types of non-DNA information as investigative tools. I know you are living in a country with a stereotype racist police problem. And I know there's lots of concern if those tools are given to racist policemen, because anecdotal knowledge, again, I don't think they are good numbers, perhaps they are, tell that if that knowledge doesn't fit the policeman's idea, he will ignore it. And if it fits, then he knew anyway. So this is useless. At least I hear those arguments from people that are critical about that, especially in the US. They may also be racist police in the Netherlands, maybe not as much. At least those policemen I talk to, they seem to use these things quite objectively, and they trust the DNA probability, and they don't really charge based on their pre-opinion. Okay, so if you want to read more, there's a review I wrote in 2015, which gives lots of details. I'm currently working on a new one that should come out end of the year. So that will have an update, will also include ancestry and age information in one review. So there's more details you can read about. And these are a long list of people, well, not so long, but many I couldn't put in, of those with whom we did all the work together. On the right-hand side, you see a picture from people in my department. Not all of them are involved in this type of research. Not all of them are there anymore. And here you have the organizations that have funded this work.